Okay. Nice to see everyone again. And today I'm going to present the chapter 17, Bioluminescence-Optogenetics. So generally say that in this chapter, we're going to talking about luminopsin fusing protein. It's a light-sensing opsin that is fused to light-emitting luciferase. And we're also talking about breadths at the bioluminescence at resonant eminent. This it's quite close, like the concept is quite close, like with the frets, but just the fluorescent is not directly from fluorescent protein, but it's from an enzyme that under some chemical reactions then provide the light. It's slightly different with the frets. The application of optogenetics is limited in two critical ways. The first, the light used to excite most opsin is highly scattered and absorbed in brain tissue. So far, majority using especially for those channel rhodopsins are blue light. And it's super hard to modify to get a mutant red shift. And in this approach, we also tried in our lab. It's really hard to achieve because if moved shift the construct, it may cause the expression problems, and even it failed to construct functional channels. So this for the deep tissue, so it's a really like applicable for using the optogenetics tools. Secondly, the light source external, necessary divert to target tissue, often through a fiber optics. So like if we need to provide light, so the animal or the human, like the patient, need to install a light device and also connect the location of the interest in the region with an optic fibers. So these are two major disadvantages for applying optogenetics in your system. So in the natural world, in the deep sea, we know that a fish called the Latin fish, it has an organ that it can emit light in the deep sea. So in this chapter, we're going to talking about bioluminescence. It's quite like this Latin fish that on the device or on the channel rhodopsin, it linked with another part, and here usually is a luciferase, is an enzyme that can hydrate the luciferin and emit the light. So in another so far, and gene therapy is quite focused is the chemogenetics, and the disadvantage under the chemogenetics is that it cannot provide precise temporal or spatial control of neural activities, because usually the drug delivery usually from IP or into the vein, IV, or intranasal through the nasal, or install a canal that's delivered. But these drugs may affect several locations of the neural systems. So it's very hard to precise control and the spatial. Also, time is also another issue, as activation of a chemical actuator depends on diffusion and clearance of the activating molecule in the brain. So all this process is very hard to control. So in this chapter, we're going to introduce the luminant opsin fusion protein. This approach solves the limitation of the light delivery to the deep brain for opsin activation, as light is generated in the vicinity of the opsin internally, obviating the need for implanted hardware. So here we're going to introduce like excitatory this luminopsin and also inhibitory the LM2. So usually it's like using the channel rhodopsin, the cation channels that are linked with a luciferase from the Gaussian luciferase, and this luciferase is coming from a deep sea creature organism, and its substrate is luciferin. So after this enzyme that uh, do some chemical reactions on the luciferins, then you emit light, and this light can be captured by a channel rhodopsin, then emit, then oh make this channel conformation change and open this channel. And inhibitory, the rhodopsin were linked to renal luciferase. So this luciferase is from another deep sea creature that's a renal that can and also use the luciferin as a substrate. So generally say that the bioluminescence of the genetics is like a linked a light resource with the channelrhodopsin. It is so close, looks like the Latin fish that it brings a Latin for itself for its own use for the light signals. And the substrate here is the CTJ covalent resin. And CTJ under the chemical reaction with the luciferase, and it will change the oxygen into carbon dioxide, and in this way to generate light. And substrate is CTJ. CTJ is quite um is safe for human beings, although it's not like endogenous that present in our body fluid. So every time for this bioluminant optogenetics works, it need to provide these CTJs induced into the biological systems. So here, so we can see that because bioluminescence is a kind of like a coupled opsin with the luciferase. So there are several different types of luciferase, G luciferase, nanolatin, and several different kinds of opsins. So plus these two together, and we can have a 2D chart that can link these two together. Like we can see like the SFLMO, it's combined with the step function, the opsin, then linked with SB group and GLUC, and linked to got this. And this is excitatory. So this chart is summarized so far, already got the bioluminant activation. So for this bioluminant molecules, it has two ways that can activate these channels. So one way is the chemogenetics approach, that this enzyme that do reaction with the luciferin, and you emit the photon can activate tryptopsin, and also without the substrate, it can also directly provide external light to activate the channel. So these two are the same construct, the same molecules can be activated by external light, similar to conventional optogenetics. So here, in this example that's published in 2020, and this FLMO, it's a step function luminance. So we can see that, so it itself, it has a yellowish, like a fluorescent that linked to, so like here, the yellowish from the EYFP as indicates the presence of the fluorescent protein on this whole bioluminescent molecule on these neuron cells, and then they can activate with the blue light, directly with the blue light. You can show this is a light response intracellular at a cation current, and it's a bistable. It can be turned off with a yellow light. In another way that can provide the subtract of the CTJ, then CTJ with under the luciferase, it can emit the fluorescent light and the same colored like this stimulation, and you can also induce the cation influx through this molecular device. So we can see here that this luminance can provide enough light intensity and photons to activate the same channel rhodopsin molecules. And so here they also compared with the different device and show that for induce the current and maximum photocurrent, and this MO3 can provide this, and to see that there's a CTJ induced current, and for this SF family, it can produce the quite high fluorescent current. And the coupling efficient ensure that it's here as much better than the first generation of MLMO. But here we need to mention that because for the provide of the substrate of the CTJ, it's this substrate is injected through the injection canal using syringe pump. And the harness need to be attached to upper body rod, and the ejection canal is inserted. So it's a kind of like because it were injected and you were installed with a canal, it's quite like the optic fiber, right? So it seems that it didn't reduce the harness device comparing to optogenetics, because it still need to like insert to the location of in this study, it's the SN. So let me see. Oh yeah, here. In this study, it's still a very deep tissue. So in this chapter, the authors also like evaluate and this bioluminescence if can like being go past away go through the skull, the bone tissue, and the skin tissues. So they do the first like one like firstly they do the IV injection to introduce these genetics molecules, and then after 15 days they do the experiment, then with the IP injection, like inject this CTJ into the animal's abdomen, then captured with the CCD. And here I find that this fluorescent light is so strong that it can be captured through the transcranial transcutaneous. That's a That's a pretty cool because this endogenous fluorescent is quite high, and this do not need to like install like inject the substrate directly at the brain tissue. It can provide with the IP. So we can see that when after injection of the CTJ after like 15 minutes, it reached to the peak. Then after 250, always like return to the baseline. And and it's pretty cool, and its fluorescence is quite strong that it can penetrate to the outside, and it can be captured by a CCD camera. And comparing them to the DREAD, the DREADDs, so they're using they also provide chemicals, but the endogenous the molecular device is also an external like the transgenic approach that it can affect with this one, and this can target it to a specific tissue, but how to activate these molecules and when, and it's hard to control. And usually for this, there are those GQ, these are G protein coupled receptors, and it's for temporary, so it's even harder to control. But however, I'm thinking that for biological reactions, maybe G proteins is better because it can have amplified the signal cascade and have a longer effect. I think that's not bad. And for the CTJs, its substrate is healthy. I mean, it's not healthy. I mean, it's at least it's a safe. But for the DIDs, the CNO, it's from the clozapine, and for the clozapine, this molecule itself, it's an antipsychotic drugs. And in another issue regarding with the CTZ is that luciferins and it's a substrate for the ABC family transporter. ABC family transporter is inserted on the plasma membrane, and it will transport this molecules, this substrate, go to out of the cells. So these activities, ABC family transporters activities may limit bioavailability for luciferins in the brain, as they also expressed at the blood-brain barrier. So it may removed those luciferins from the region of interest tissue. The CTJ is a natural antioxidant, and it's quite safe. And excessive higher dose an oral of this CTJs shows no sign of acute chronic toxicities. And it have many ways to delivery, but it still need to dissolve in a solvent like DMSO first before diluted into aqueous solutions. And this provide with have a various routes, including intra IP like IV or intracortical or the intranasal. Intranasal delivery is a specific. And it's very convenient, especially like for the CTJ, and because there's no blood-brain barrier, and in the nasal, these approaches. So generally, to compare the time course of the optogenetics and bioluminescence-optogenetics and chemokines, the optogenetics is quite sharp. Get turned on, turned off, quite sharp, quite fast. And in the middle, the time course is the bioluminescence-optogenetics that we introduced today. For the chemical genetics, is much more slower, but it can prolong longer. So in this table is summarized between different approaches. Like optogenetics trigger is using the light, and the chemical is chemical genesis chemical, and bioluminescent is candle light, or with the breaths. And it need to provide light, and for a chemical we provide different substrate, different drugs molecules. And the mechanical is entropic, metatropic, entropic. And the region of operations of the gen is small, and chemical can be several locations and brain wide and several places. So it's a variable. And that's also with the bioluminescent of the genetics. For the kinetics of genetics fast, chemical genetics is slow, and bioluminescence and need to dependent on which luciferase is using and which rhodopsin is using. An off-target effect of genetics is quite low, but the chemical is possible. Just like we mentioned, CNO, the clozapine different, and it's possible that they have off-target effects. Intrinsic active monitor infer from light intensity and opaque of tissue. Chemical genesis have no, and bioluminescence is intrinsic monitoring. So here we can introduce some of the applications. So here in this 2018 publication, it's to treat the epilepsy. That we can see here with provide this the C-force is dramatically reduced compared to the controls. So it shows that it can reduce the acute seizure. So with this bioluminescence-Optogenetics tools, so this is for treatment with the epilepsy. And another publication is a treatment for the Parkinson disease. That's first. So here they used combine several techniques, of including this MMO, this bioluminescence system, and also with the stem cell therapies. So first, they transfect some pre-differentiate like stem cells and with this system, and after you start to differentiate into some progenitor, inject into some brain regions, then this region, so these molecules and those stem cells, after transplant into striatum of mutant mice, and it will chronically delete and shows a recovery of the motor functions. So this animal is a Parkinson disease animal model that it shows Parkinson disease, those phenomena. So those implanted like transgenic stem cells, these neuron precursors can shows positive benefits of this Parkinson disease animal models. And without the daily injection of the substrate CTCs, and animals first to demonstrate a recovery of motor functions. So here is another application for the bioluminescence driven optogenetics activation. So overall, it gives a deep tissue light luminance without fiber optic fiber installation on here. So this is the major concept of this system. Thank you.